These are the lecture notes for Chapter 3. This is also a chemistry chapter, but it is going to focus on the chemistry of organic molecules, and that means that these are the 4 primary molecules that are found in living things, and all of them contain carbon and hydrogen together. So, excuse me. So this is a list of, first there's, in section 3.1, there's a section on basically what it means to be organic, and then the other 4 sections are on the 4 biomolecules or macromolecules. They're the 4 molecules of living things. They're carbohydrates, lipids, proteins, and nucleic acids. So starting with section 3.1, it's important that you know that if it's organic, a molecule will have both carbon and hydrogen. Carbon and hydrogen. If the molecule only has carbon or only has hydrogen, it doesn't count as organic. And then the classes of biomolecules are listed again: carbohydrates, which we shorten and say carbs a lot, lipids, proteins, and nucleic acids. This table lists functional groups that you might find in organic molecules. Hydroxyl means that the functional group contains an OH bonded to another chemical. The R just stands for whatever chemical that it's bonded to. A carbonyl group is C double bonded to O, single bonded to H. A ketone is going to be a carbon double bonded to an oxygen and then bonded on either side to other chemicals. An amino group, we'll definitely see amino group, which is a nitrogen bonded to 2 hydrogens. Phosphate group for sure, because we see phosphate groups in DNA. So that's going to be a phosphorus surrounded by oxygen atoms. Okay, so carbon is found in living things because one of the most important reasons is it can form up to 4 covalent bonds. So carbon is satisfied when it forms 4 chemical bonds where it shares electrons with 4 other substances. And then I'm just moving on. When we talk about these different groups, these different biomolecules or macromolecules, you need to make sure you're familiar with the terms monomer and polymer. So if these are very long chains, these molecules are very long chains, then the individual link in the chain is called a monomer, and then the chain itself is called a polymer. So for example, amino acids are the monomers. They're joined together to form a chain called a protein. So the protein would be the polymer, the amino acid would be the monomer. So monomers are really just building blocks. And this table is important because it tells you for each of the 4 categories what the subunits or monomers are, and then what the polymers are called. So if it's a carbohydrate, the building blocks or monomers are called monosaccharides, like glucose. You may be familiar with glucose because it is the simple sugar that's found dissolved in our blood. We call it blood sugar. So glucose is a monosaccharide, and chains of glucose would form a polysaccharide. Lipids do not have an asterisk because lipids don't technically form polymers, although they can be very large molecules. But the subunits of lipids are glycerol and fatty acids. And fatty acids do consist of long hydrocarbon chains, so chains of carbon and hydrogen. Proteins, the monomers are amino acids, and the polymer is actually called a polypeptide. It's going to be called a polypeptide until it undergoes certain shape changes that make it into the final 3-dimensional structure of the protein. But polypeptides and proteins contain the same amino acids or the same building blocks. So sometimes you'll see amino acid as the monomer and the polymer either as polypeptide or protein. And then for nucleic acids like DNA, the monomer is the nucleotide, the polymer is either DNA or RNA. Now, a dehydration reaction is a chemical reaction where a larger molecule is synthesized. So dehydration reaction is used to connect monomers together to make polymers. So for example, the formation of starch, which is a polymer, it's a polysaccharide polymer from glucose subunits. So glucose would be the monomer and the polymer would be starch. And the reaction that joined the glucose monomers together is called a dehydration reaction. Sometimes it's called a dehydration synthesis reaction. So synthesis means to build something larger. The breakdown reaction that we see with organic molecules is called hydrolysis. Lysis means breakdown. So hydrolysis reactions are used to break down polymers into monomers. For example, when we digest starch, so if we eat a piece of bread or a potato, we have enzymes that start breaking down that starch into its glucose monomers, and that would be an example of a hydrolysis reaction. You can see here that dehydration reaction is used to join two monomers, and the reason for the word dehydration is because one of the monomers, the monomer on the left, has an OH attached to it. The monomer on the right has an H attached to it. So if you remove the H and the OH, then you leave room for those two monomers to join, and then the H and the OH form water, H2O. So it's called dehydration because water is removed during the reaction, and it's a dehydration synthesis because you are building a larger molecule from two smaller ones. Two monomers join to make a larger molecule. That's going to be dehydration synthesis. And then hydrolysis is when this larger molecule formed from two monomers, when it splits by adding the water back. So the OH is added to the monomer on the left and the H is added to the monomer on the right. And so hydrolysis is basically the reverse of the dehydration. Here's a picture of both reactions. In the dehydration reaction, you have two small monomers that are joined to form a larger polymer, and you also have the removal of water. In the hydrolysis reaction, you start at the bottom. The two monomers are split. The molecule that's made of two monomers is split into the two individual monomers, and water is added back. So dehydration and hydrolysis, just make sure that you know the purpose of each one. Dehydration is for synthesizing larger molecules, and hydrolysis is for breaking down those large molecules into their monomers. Okay, enzymes are protein molecules, so they're always proteins, and they speed up a chemical reaction. So down at the bottom, the word for that is enzymes are catalyst. So a catalyst is a substance that speeds up a chemical reaction. Enzymes are not consumed in the reaction and they're not changed by the reaction. So basically, if it's an enzyme that helps to break down starch into glucose, the enzyme can be used over and over and over again to do that. All right, the section on carbohydrates. Carbohydrate, the word itself tells you the elements that are found in carbohydrates. Carbo for carbon, hydra for hydrogen, and ate, A-T-E, on the end of a chemical word means that there is oxygen involved. So the functions of carbohydrates are for an energy source. They give us energy. And they provide a structural role in the body. So carbohydrates also provide a structural role. And carbohydrates, particularly the smaller monosaccharides, Simple sugars are going to contain carbon, hydrogen, and oxygen in a special ratio, a 1 to 2 to 1 ratio. So glucose, for example, is 6 carbons, 12 hydrogens, and 6 oxygens. So 6 to 12 to 6, which is 1 to 2 to 1 when you reduce it. The varieties of carbohydrates are monosaccharides, disaccharides, and polysaccharides, and you'll need to know some examples of each. Here's a picture of a cactus. The cell walls of the cactus contain a polysaccharide called cellulose. The shell of this crab contains a polysaccharide called chitin, and the cell wall of this bacterium contains peptidoglycan. All right, monosaccharides are also known as simple sugars. They tend to have 3 to 7 carbon atoms. And remember, if they've got 3 carbon atoms, then they have 6 hydrogen and 3 oxygen. Glucose is blood sugar, fructose is fruit sugar, and galactose is found in milk. These are 3 monosaccharides that have 6 carbon atoms each, so we call them hexoses. And then there are 2 pentoses with 5 carbon atoms. They are ribose and deoxyribose, and these are found in DNA and RNA. This is a picture of different ways to represent the molecule glucose. You will need to know this. You will need to know C6H12O6 is the chemical formula for glucose. The other images are just showing you that glucose tends to be drawn as a green hexagon. Now, disaccharides contain 2 monosaccharides joined together by dehydration synthesis. The examples that are listed here are lactose, sucrose, and maltose. Lactose is a disaccharide composed of the monosaccharides galactose and glucose. Sucrose, or table sugar, is composed of glucose and fructose. And maltose is composed of 2 glucose molecules. People who are lactose intolerant lack the enzyme lactase, which breaks down lactose into its 2 monosaccharides, galactose and glucose. So they can take that over the counter, and it will help them to digest milk, the lactose in milk. This is showing you a hydrolysis reaction where maltose is our disaccharide, and it is being split up into its 2 glucose monomers. And then polysaccharides are going to be chains of monosaccharides. Examples are starch, glycogen, cellulose, chitin, and peptidoglycan. Starch is the storage form of glucose in plants. Glycogen is the storage form of glucose in animals. Particularly our liver cells and our muscle cells will store glucose in long chains called glycogen. Cellulose is found in plant cell walls, and animals such as humans are unable to digest cellulose, but some animals have special protozoans that live inside of their gut that help them to digest it, like, for example, termites. Chitin is found in the cell walls of fungi and the exoskeleton of animals like crabs, and peptidoglycan is found in the cell walls of bacteria. You can see here a picture of image of the starch found in this potato, long chains of glucose, glycogen found in this liver, long chains of glucose. They look very similar. And cellulose found in plant cell walls, again, is long chains of glucose. And now we're on the section called lipids.